Hello, this is Gina Piscatelli with a pre-recorded lecture for information on our eye physiology lab for Anatomy and Physiology 1. In this presentation, I'll talk a little bit about the retina and how information is sent to the retina and then the optic nerve. And I'll also discuss how we use both eyes to view distant objects and near objects. This will involve 4 separate topics that you can find in your lab manual on page 139. The topics include photoreceptors and their function, the function of the lens, how it bends light waves, and the term for that is refraction, the bending of light waves. And then the third topic has to do with the function of the iris in controlling the size of the pupil. This is done to accommodate the different light intensities we experience when we view objects, as well as how far away objects are or close. And then the fourth topic is more anatomy than it is physiology, but it does relate to some function, and that is the visual pathway all the way to the brain, from the eye to the brain. So I recommend that you have your lab manual on hand while you're watching this so that you can take notes on certain pages as I discuss these 4 different topics. So first I'm going to start with the first learning objective, the types of photoreceptors in the retina. This is review for most of you. We've already talked about rods and cones, and that the fact that rods are stimulated by white light. We perceive this white light in shades of gray, dark to light. And we've talked about cones and that they are stimulated by colored light or wavelengths that we perceive in color. However, something that wasn't mentioned is that there are really 3 types of cones in the retina. Some cones are stimulated by wavelengths that we see as blue, and other cones are stimulated by wavelengths of light that we perceive as green. And then finally, the third type of cone is stimulated by wavelengths of light that we see as red. Okay, but together, the rods and the cones give us excellent visual acuity. We're really able to discern fine detail. Now, these cells are not actually neurons, although they do release neurotransmitters. Often they're referred to as neuroepithelial cells or just photoreceptors. Neuroepithelial refers to the fact that they look like epithelial cells, columnar shaped, and they release neurotransmitters. Now, how are they stimulated? Well, they contain pigments in the outer segment. So in this region right here, the outer segment, which is the most peripheral of the retina, and I'll show you a picture of that in a second, contain these pigments that change shape. They absorb light and they change shape. These pigments are often referred to as opsins. You may have heard of opsins. So this is really where stimulation occurs, in this outer segment. Okay. Now when the pigment changes shape, it will cause ions to either enter or maybe exit. It depends on whether or not an ion channel has been opened or closed. But the result is activation or deactivation, depending on what we're looking at. Now you have this picture on page 140, and it just shows you or reminds you that the area of the retina that has the highest density of cones is the fovea centralis right here. So when light rays hit hit this part of the retina, the most photoreceptors are stimulated, but that would be for in bright conditions, because we would see colored objects. In the dark, when we're trying to view shades of gray, it's better when light hits other regions of the retina, because that's where the rods are located. A region that doesn't have any photoreceptors at all is called the optic disc. So if light rays come in and hit this part of the back of the eye, we can't see anything from that region of the field of view that the light rays came from. We usually abbreviate field of view as FOV. So if a light ray has bounced off an object in this location so that it hits right at the optic disc, it creates a blind spot in your vision, in your field of view. Okay, let's look at the retina in a little bit more detail to finish off this first learning outcome or objective. This picture you don't have in your lab manual, but you have the more detailed one on page 145 of your lab manual. So I'll give you a second to turn to that page. You're going to want to draw on it in a second. So the detailed larger picture comes from the back of the eye, and it shows right here is the back of the retina. And light enters the eye anteriorly. So this more detailed picture that you have shows light coming in anteriorly, and yet the photoreceptors, the rods and the cones, are very posterior. So the light has to penetrate layers of cells in the retina in order to reach the rods and the cone pigments, the visual pigments, at the very deepest part of the retina. Now, these cells have names that the light has to penetrate. Most anteriorly is the ganglion cell layer, and then there's this layer of neurons called bipolar cells, and then finally you have the photoreceptors. So I have another picture that I like better. I think it's better. It's brighter, at least. And it shows the eye in a position that I think is a little bit more intuitive. And that is with the pupil facing up, and back here is the retina. So light will come in from the anterior region again, and on this bigger picture, light's coming in from this direction. And, I guess I draw it right here. Notice how many cells the light waves have to go through to find the rods and the cones. It's kind of interesting. So the ganglion cell and the bipolar cell types, they do not respond to the light waves. Only the rods and the cones do. But the rods and the cones are going to communicate with the next cell layer, and that will be the bipolar cell. So the rods and the cones synapse on or communicate with the bipolar cell through a neurotransmitter, and the bipolar cells communicate with a ganglion cell or two with a neurotransmitter. So the first take-home message for all of this is just that light has to penetrate layers of the retina and reach the most posterior part of the retina, or the deepest layer of the retina. That's the first take-home message. The second take-home message has to do with the path that light travels after the photoreceptor, the rods and the cones, have been stimulated. So we have the rods and the cones really deep, and they are stimulated, and they communicate then with the bipolar cell. Well, now the signal, the neural signal, is moving forward anteriorly, as is true from the bipolar cell to the ganglion cell. So this information, first the light rays came in from the anterior to posterior, and now the neural signal is going in the opposite direction. The ganglion cells have these axons that then travel, join other axons, travel to the optic nerve, and now the optic nerve carries the neural signal posteriorly again. So this optic nerve will move posteriorly, and we'll talk about how it gets to the visual cortex in the occipital lobe of the brain a little bit later. So I recommend, just for ease of, because you don't have that same picture that I have, for ease of drawing, that you add some information to the picture on page 145 that you do have. Make sure you notice where the light rays are coming from the anterior. So this is the anterior of the eye. And the light has to penetrate one layer, two layers, and then get to the rods and the cones, the outer segment, I guess, of the rods and the cones in the retina. Now they're stimulated, they're excited or inhibited, it depends on what you're looking at. And now a nervous signal or neural signal travels towards the anterior part of the eye. And finally, the ganglion cells have an axon that travel to the optic nerve, which goes to the posterior side of the eye, or the back of the eye, comes off of the back of the eye. So I just draw in these arrows in my lab manual to show the direction of the neural signal. Again, the neural signal starts at the rods and the cones. It moves anteriorly, travels kind of laterally to the optic nerve, and then it moves posterior in the optic nerve. Kind of interesting. Okay, we're going to move on to part 2 now. And this deals with the lens, how the lens functions. The purpose of the lens is to help us see objects near and objects that are far away. What it does is it refracts light rays, so it bends light rays. And the purpose of this, bending the light rays, is so that the light rays converge within the eye and fall directly on the retina so that we get a focused image, not blurry. Now, the bending of light rays needs to vary depending on whether you're looking at something near or far. And the lens is able to change shape so that it bends light rays different amounts, depending if you're looking at something far away or near. So the ability of the lens to change its shape is called lens accommodation, so that we can focus on both objects that are far apart, far away from us, or near to us. This is a picture you don't have in your lab manual. I wish you did, but it shows you why refraction occurs. As light rays travel in one medium, they travel at a continuous speed. But as soon as they hit an object of a different kind of material, like this glass, they change speed. Typically, in this case, slow down. And when they slow down, they bend. So, or they're refracted, we say. So we have a point of refraction when the light rays enter the glass, but then the light rays reach the air again and they speed up. So the light rays are bent again. So this just shows you that refraction occurs when light rays hit any new material because they change speed. So we have two different structures in the eye that can perform refraction, bend light rays. One is the cornea and the other is the lens. The cornea is static. It never changes the amount that it bends the light rays. Always the same angle, always. And that's because it doesn't change in thickness or consistency or anything. It's pretty static, except with age, but that's another story. The lens, however, also performs refraction, and it does have the ability to change the degree of refraction. Okay, so it does that because its material is flexible enough to kind of stretch and then ball up, stretch and then ball up. So we're going to look at a picture here that shows you the case on the left here. It's not the left eye, but boy, this isn't working too good. That's supposed to say left. Doesn't look like left. But it shows you the lens is pretty thin if you look at the diameter. Versus the picture on the right, it shows the lens is thicker. And notice that a thin lens doesn't bend the light rays very much. The angle, whatever the degree is, I don't really know, of the light ray as it moves through the lens gets bent just a certain amount. But in the thicker lens, the angle of refraction or bending is greater. The light ray comes in and it's bent more. So when the lens is thin, light rays are bent less. We use this to see objects that are far from us. So this is the condition of our lens when we're viewing far objects. Versus when the lens is thicker, light rays are bent more, and this is so that we can bring near objects in focus. The next picture here you do have in your lab manual. It's on page 141. It shows you two conditions of the shape of the lens. Here the lens is thinner on the left, and on the right the lens is thicker. Now this means, of course, that the light rays that enter the one on the right will get bent more. So I'll just draw a light ray in here. Comes in and it gets bent quite a bit. The light ray that comes in at the thin lens, it won't be bent quite as much. Okay? Now, how is that achieved? The purpose of today is really to talk about how it is achieved that the lens can change this thickness or shape. It occurs due to the action of the ciliary body, which is made up of smooth muscle. So the smooth muscle of the ciliary body is shown in pink right up here underneath the iris. So the iris is that first pink line. But deeper is the ciliary muscle. When the ciliary body is relaxed, that ciliary body actually kind of rolls backwards. Okay, it relaxes in a reclined position, if you will. So there's a little green arrow that shows the ciliary body rolling backwards. Essentially what this does is make the thickness of the ciliary body, thinner. The width, I guess we'll call it. It's thinner. When this muscle is relaxed, though, notice that it has suspensory ligaments that are attached to the lens. So it rolls back, the suspensory ligaments get pulled tight or taut, and that makes the lens flatten out. They pull on the lens and the lens gets thinner. So relaxation of the ciliary body makes the lens thin. On the other hand, if the ciliary body contracts, it kind of rolls forward. And this is a picture of an anterior view below of the ciliary body, and it shows that it kind of gets thicker, it widens. The result of this is that the suspensory ligaments become more slack, and therefore the lens isn't pulled on, and the lens can kind of round up into its natural shape. It becomes more globular, rounder, and thicker. This is the picture, the next picture I want you to focus on. It's back on page 141. And it shows you that when we have a thick lens, the rays get bent more than if we have a thin lens. Okay? And I'm bringing that up because right now we're going to talk about when we need the lens to be thick and when we need it to be thin. So in order to view objects that are close to us, we're going to need to bend the light rays a lot. I need to use a different color here. Go with purple. So we need to bend light rays a lot more. And if you notice, that's because distant objects appear smaller. The light rays come in really close together. They're close together as they enter the pupil. So to get them to converge inside the eye, they don't have to be bent as much. But when we look at objects that are close to us, they appear bigger, and the light rays enter farther apart. And in order for them to converge, they're going to be inside the eye. They're going to need to be bent a lot more. And so you have the point of conversion or convergence inside the eye. Okay, and then the rays eventually fall on the back of the retina to form an inverted image, an upside down image. So we need the lens to be thick to bend the light rays a lot to see distant objects. Near objects. So we need a thick lens to see near objects. And to get a thick lens, we need the ciliary muscle to contract. So the ciliary muscle contracts, the lens thickens, and now we can see a near object. But of course, in the periphery of our vision, there are distant objects. Those objects will appear blurry because notice how far apart the rays fall on the retina. You don't get a nice fine focused image on the back of the retina when you have a thick lens. Now let's look at what our lens does for far vision, and of course it's going to be the opposite, right? So distant objects again appear smaller. The rays come in closer together than do from a near object. And so we don't need to bend them very much to get convergence. So what happens in this case is the ciliary muscle relaxes. The suspensory ligaments are pulled tight, and the lens flattens or becomes thinner. When it becomes thinner, the rays are bent less, but they still converge just fine because they're so close together already. And they fall on the retina kind of close together to get a nice, sharp, inverted image. Now, what happens to the near objects? Well, the rays come in from near objects, far apart still. We've got this thin lens, but they don't get bent. These rays don't get bent enough, so they don't even converge right in the eyeball at all. And so they will appear blurry, generally speaking. Now, it is true that within the eye, light rays are able to bounce around a little bit, and so you might get some photoreceptors excited, but it'll be so scattered that the image will be blurry. It won't work. So this brings up something I wanted to mention from the beginning and forgot to, and that is that when we look at objects with a certain orientation, like this flower, the light rays bounce off of the object and enter our eye. And because they're reversing direction, the focused image on the retina is inverted. It's inverted. So it's upside down or backwards, depending on the orientation. That's what I mean when I say inverted image. Okay, so now let's talk about visual impairments that relate to lens function, or at least to refraction. There's 3 different visual impairments described in your lab manual. One is called presbyopia. This is due to the aging process, and the lens loses its elasticity and it can't accommodate very well. So it just happens with time. And it's usually easier to see near objects than it is far objects, but it can change. I'm sorry, far objects versus near objects, but it can change over time. We aren't going to discuss how that's corrected very much. Usually you get bifocals, but that's for another course, I think. For us, what we want to look at are 2 conditions that are pretty common in younger people, younger adults, and children. That's myopia and hyperopia. Myopia is often referred to as nearsightedness. Hyperopia is referred to as farsightedness. So when you're nearsighted, that means that you can focus on objects that are near, but distant objects aren't focused onto the retina sharply. And that's usually because the rays converge too far or too close to the lens. So the cause of this can be that you have a thick lens that's not very good at accommodating and becoming thinner, or you have an eyeball that's too long. Now, how do we fix it? Well, we use a lens in front of our eye. We add a lens to the equation, and we use a concave lens that's, well, I can't draw it very well here. We'll go back, we'll go to the next page and you'll see it in a second. Hyperopia is the opposite. Near objects are not focused in the eyeball. Convergence doesn't even happen in the eyeball. Now, this is usually because the lens is too thin, but it can also be that the eyeball is too short. So this is just something that has some genetic predisposition typically. So in order to fix this, we need to add a lens that bends the light rays more. And so we add a corrective convex lens in front of the eye. So let's review the different types of corrective lenses just for shape, since I can't draw very well with this. A concave lens that we use to treat myopia has this curved surface inward. It's caved in on both sides. And when light rays pass through it, they're refracted They go through it and they're refracted away from each other, kind of. They're spread out. Versus a convex lens, that's what we use to treat hyperopia, and it's curved outwards on both sides. The light rays enter. This is thicker where most of the light rays enter, and so light rays are bent more. Converges the light rays. So I'm going to show you an example here. For myopia, either the eye is too long, as in the top picture, or the lens is too thick. It's very globular. And so that means is that we can see near objects just fine. The rays are bent quite a bit. Convergence happens in the eye. You can see a near object. But if you're trying to look at a distant object, like somebody that's really far away, you get convergence too far anteriorly within the eye. And so the light rays, when they spread apart again to hit the retina, are spread quite far apart, and you get kind of a blurry inverted image on the retina. The way we correct this is with a concave lens, and the light rays come into the concave lens and they are spread apart a little bit first, and then bending happens. And bending will happen a little bit farther back. I mean, convergence will happen a little farther back, and so you'll get a nice sharply focused image on the back of the eye, in the retina. For hyperopia, it's the opposite. Either the eye is too short or the lens is too thin. So in this case, the rays aren't bent very much and convergence happens farther back. To correct this, we use a convex lens so that bending happens more anteriorly. Okay, so that's it. We're going to do an exercise on that later, but that's it for learning objective 2. Now for learning objective 3, we're going to talk about the iris and the pupil. The iris is also composed of smooth muscle, like the ciliary body is. And it controls the diameter of the pupil. In order to change the number of light rays that enter the eye. Now you can imagine if you're in really bright light, the pupil's going to constrict, right? Because you don't need all the rays that are available to see what you need to see. In fact, your vision would be bleached out a little bit. But in conditions that are darker, our pupil dilates so that we can capture more light rays and see what's present in the darker environment. So how does the iris change the pupil size? It consists of 2 smooth muscle layers, really. A circular muscle that's kind of a sphincter muscle, we call it. That's on the inside. And then radial muscle, which is kind of radiating outward on the periphery. So the one that's in the center is called the pupillary constrictor, because when it contracts, the pupil is constricted. And that's shown on the left here. We've got the circular muscle contracted. You have this figure on page 143. And in the picture on the right, we have the dilated pupil, and that's due to the radial muscle of the iris contracting. And so the iris is brought further away from the center and the pupil dilates. So the result of the pupil being able to dilate or constrict helps us with not only limiting or expanding the amount of light rays, but it also helps us see objects that are near versus far. This is because objects that are close to us bounce more rays into our eyes. More light rays come off of near objects and enter our eyes. So it could be too many, right? So the pupil constricts in order to limit, because otherwise the near object would appear way too bright. But in the case of distant objects that are far away, fewer light rays enter the eye than near objects, and they would appear too dim unless we could dilate the pupil. So with this accommodation performed by the iris and the pupil, constriction of the pupil allows us to see some detail of near objects without too much brightness, and dilation of the pupil allows us to see distant objects, because then we capture enough light rays to see distant objects. So, summary of the lens and pupil accommodation for far versus near objects is that for distant objects that are far away, the pupil is dilated, the ciliary muscle is relaxed so that the lens is thin. For near objects, the pupil is constricted and the ciliary muscle has contracted so that the lens is thicker, and that means that light rays will be bent more as they come in. So that's just a summary. Okay, let's do an activity that you have on page 151. On page 151 is a activity listed as number 8. Determine corrections for myopia and hyperopia. So in normal vision, we typically have the image forming on the retina at the back of the eye. The lens is the right shape to get convergence where we need it. And in the case of somebody who is nearsighted, the image doesn't fall on the retina. Instead, the image is produced really in the vitreous humor. And that's because the light rays, the light rays are actually bent too much. So how do we fix myopia? What type of lens would help? The answer would be concave. Okay, that's writing isn't going to work, but you get the idea. And then in the case of farsightedness, somebody that's farsighted, hyperopia, what happens is that the image doesn't fall sharply on the retina at all, and so the convergence happens too far posteriorly. The image actually falls behind the eye. And so in this case, we need to bend the light rays more, so we use a convex lens. I'll try to draw a convex lens in here. So if you have any questions about what the answer should be for this, the next slide does have the answers, and I've posted these slides for you. So that was our third learning objective. The main purpose is to show you that the ciliary body controls the shape of the lens and the iris controls the size of the pupil. Now let's do learning objective number 4. This is anatomy of the visual pathway from the eye all the way to the part of the cerebral cortex that helps us perceive what we're seeing, right? So we have light rays that enter the eye, and the retina responds, is stimulated. And then the information travels in the optic nerve all the way back to this structure called the optic chiasm. From the optic chiasm, there are electrical signals or neural signals sent by the optic tract all the way to the occipital visual cortex in the back here. Okay. Now, you may have heard that really visual information is kind of separated in the brain, right side versus the left side a little bit. And that happens because of some anatomical pathways of neurons, and the neurons cross here in this optic chiasm. So we're going to look at how that happens next. You have this figure on page 145, and it shows you, it's on the bottom of page 145, it shows you the occipital visual cortex down at the bottom has two different color schemes. One is orange or salmon colored, and one is more green. This reflects where the image is in your field of view, or where the object is in your field of view. So this is a superior view. So it's actually probably an inferior view, but it doesn't matter. The point is that the field of view on one side, whatever is over here, be it a tree, a bush, a ball, whatever it is that's over on the right side of the field of view, will all cross over to the opposite side of the brain. The same is true for anything that's in the left field of view, but it'll go to the right side of the brain. So how is this achieved? Well, notice that if I use this color scheme for the field of view, that within the optic nerves, we have some neural signals that are color-coded both green and salmon. So what that means is the optic nerves carry signals from both sides of the field of view, the whole range. Both eyes and both optic nerves are able to capture visual information from the entire field of view. But it's at the optic chiasm where some information is going to cross over, so that the optic tract that leads away from the optic chiasm will only carry information from the opposite field of view. So notice on the left-hand side of this picture, we only have salmon-colored neurons traveling in the optic tracts, and that all came from the right field of view. So it's really the optic chiasm that helps this, and the pathway of the axons or the neurons. Okay, so what's most important is this, and you have this as well on page 146, is that to understand this, you really want to think about why this occurs. And I'm going to trace the pathway, and then we're going to do it together so that you can understand why some information crosses and why some information doesn't. Okay, so I'm going to use blue because that'll be easier. So this is a superior view, so we know what's left or right. And in the field of view, left is pink and right is green. Now, light rays from the left field of view, I've drawn as pink in your diagram, and they're going to enter both eyes, the left eye as well as the right eye. So we have some pink colored left rays coming into both eyes. But notice they come in at a certain angle. From the left, they kind of are diagonal into the right side of the eye. So the right side of the retina is stimulated. Now let's look and see what happens from the right field of view. Okay, so from the right field of view, we have these green rays coming into both eyes from the right field of view to the left side of the eye. That right away sets up the pathway of neural transmission and what side of the brain it's going to go to. So notice that anything in the medial eye, so for the left eye, that would be the right side of it, right here. And for the right eye, the medial part of it, which would be the left side, that information is going to cross over to the opposite side. Notice from the left field of view, we have pink rays on the right eye, and it crosses in the optic chiasm. The same is true from the right eye, the medial side here. We have these green rays that cross over to the opposite side. This may or may not make sense to you, but it tells you that if you had some sort of accident that caused the optic nerve to be cut, which is a terrible thought, you would still be able to view your entire field of view with only one eye. You might have to turn your head, but you could. And you would be able to perceive both fields of view, because eventually both fields of view would reach the brain. So you would know what you're looking at. But what would happen instead if you had some sort of tumor or accident? Sometimes it's a pituitary gland problem, but we won't get into that too much. What would happen instead if there was damage to the right optic tract, for example? Well, you're still taking in information from both eyes, but anything from the left field of view will not be perceived by the brain. You'd have to really move your head and put everything in the right field of view for you to even perceive that something is there, because you'd need it to be going to the left visual cortex. It's kind of interesting. So just to practice understanding that, you have this activity on page 152, and it's a highly schematic diagram which asks you to draw the light rays from the left field of view into the eye, through the optic nerve, through the optic chiasm, and to the visual cortex, and to do the same from the right eye. The answer is on the next page, but if you want me to do it with you again, I can do it with you on this blank sheet. It's up to you. You can stop now and try it on your own and just look at the answer. But for those of you that want to go through it kind of pseudo live again, I'm going to do it again here. So from the left field of view, rays enter at a diagonal into both eyes, which means that the left eye medial side is stimulated and the right eye's lateral side is stimulated. Now let's do rays from the right field of view. From the right field of view, rays come in diagonally to both eyes. Waiting for it to catch up. Okay, and so in the left eye, the lateral part is stimulated, but in the right eye, the medial part is stimulated. Now from this point, if you've done that part right, you'll be good, because now all you have to remember is that anything that's medial in the eyes crosses. So we'll start with the left eye. On the left eye, what's medial is blue. It's from the left field of view, and that's going to cross over to the right cortex. So from the left field of view through the left eye, it ends up on the right side of the brain. And from the right field of view, we have visual information stimulating the medial part of the right eye, so that is going to cross over the optic chiasm to the left visual cortex. So again, from the right field of view, information entered the right eye, but it went to the left side of the brain. However, there's some information that doesn't cross. For example, from the right field of view, that stimulated the lateral part of the left eye. So that information will stay on the left side. So right field of view still went to the left side of the brain through the left eye. And in the case of the right eye, it was the left field of view that stimulated the lateral part. And that information just travels straight back from the right eye to the same side visual cortex, the right visual cortex. Okay, that's all for eye physiology. There's more exercises that you can do in your lab manual if you're interested, like your blind spot, your visual acuity, things like that, but you're not responsible for doing so. And I thank you very much for your attention.